Hi guys, so I'm back again. I'm back again with a new topic today. Thank you so much for all your support. I really feel encouraging to continue my work. Today I am back with a new topic, mutation and the human genetic diseases. I would like to mention this session and the next few sessions are really very important because it's been long that I really wanted to speak about it, and good that I started all this session I am creating. I definitely want to make a social awareness for the genetic diseases. So let me start about mutation first. So mutation is something without which the evolution of species would not have been possible. The continuous process of mutation and natural selection improves the properties or features of a species. Mutation is very important for improving any enzyme properties. This mutation adds several benefits to us, to our life, gives certain attractive features. This mutation not only have a brighter side, but it has also a very dark side. So you will understand the dark side when I speak about the human genetic diseases. Also, definitely we are going to speak about the positive side of mutation when we'll go into the synthetic biology and understand how this mutation can help us in enzyme engineering and also in antibody engineering. A mutation can be inherited or it can be acquired. When a person gets some mutation from its parents, so it is called inherited mutation. But at the same time, a person can acquire a mutation during its lifetime due to several reasons. So if the mutation occurs in the germline cells, that is the sperm or the egg cell, then definitely this is going to be inherited to the offspring. But if the mutation occurs to somatic cell, so it cannot be transferred to the next generation. So it will be confined to his lifetime only. So let's understand more about mutation. So before going to deep into the subject, so I would like to give you some basic background that will help you to understand the genetic of the diseases. So guys, let us go into the slides to understand mutation, then we will speak again. So in sexual mode of reproduction in the humans, so the male produces the male gamete called the sperm, and the female produces the female gamete called the ovum. Because these gametes are produced in the process of meiosis, hence they contain haploid set of chromosome, which is represented by N. So life begins when the egg is fertilized by the sperm and it then forms the zygote. The zygote is now diploid because it has 2n set of chromosome. So it will have total 23 pairs of chromosome, and each parents contribute one set of chromosome. Now the zygote undergoes a process of division, cell differentiation, and it gradually forms an embryo, and the embryo finally forms the human baby. So the cellular differentiation occurs when the cell divides and forms different kind of cells. Differentiation dramatically changes a cell's size, its morphology, its membrane potential, metabolic activity, as well as responsiveness to signals. Based on the type of organs or the based on the function it will be doing, so it gets differential gene expression compared to other cells, and that gives it new features. Now let us look into the cell where exactly the DNA and chromosome we can found. Like as you might be knowing the structure of the cell, this is we are presenting a animal cell, and here you can see the chromosomes inside the nucleus.
And these chromosomes contains the DNA. The DNA, which is a double helical structure, it is wrapped around histone protein to form nucleosome complex, and they are tightly pegged inside the chromosome. Some stretch up DNA which can code for a protein, so that is called as a gene. Here represent the human karyotype. That means that, like, a karyotype is an individual's complete set of chromosome. So the karyotype is, for every species it is very specific. Humans typically, they contain 22 pairs of autosomes and one pair of sex chromosome. So the sex chromosome in case of female it is X and X, and in case of male it will be X and Y. So the karyotype is useful for studying chromosomal abnormalities. So if there is any structural change in chromosome or in the number of chromosome, so that is useful during prenatal diagnostics, also for tumor studies. It can define cellular function, and also it helps us in understanding the evolutionary history. So let us understand how sex is determined in humans. As you know, in humans, the male has one X chromosome and one Y chromosome, and in female, the sex chromosomes are X and X. So that means the male can produce two types of gametes. One is carrying X chromosome and another is carrying Y chromosome. Whereas the female can produce only single type of gametes which will be carrying the X chromosome. So if the sperm carrying X chromosome is fertilizing the egg, so that means the baby will have X and XX, the sex chromosome, and that will indicate a baby girl. So if a Y carrying sperm is fertilizing a egg, so that means the baby will have the sex chromosome as Y and X, so that means it will be a baby boy. So let us understand the structure of DNA. So as you know, the DNA is double helix, and if you look into the structure, so the DNA backbone is made up of the deoxyribose sugar and the phosphate group, and this phosphate group provides the DNA the acidic property, and it has four type of nitrogenous bases, that is adenine, cytosine, thymine, and guanine. In case of RNA, the thymine is replaced by uracil. And if you talk about the bonding, so the thymine pairs with adenine by a two hydrogen bond, whereas the guanine bonds with cytosine by three hydrogen bond. So there are a total 20 type of amino acids present in all living organism, and I have shown the abbreviation of each amino acid both in three letter form and single letter form. Now let us understand the codon table. So the relationship among amino acids and the codon is called the genetic code, which is often summarized as a table called codon table. So the genetic code is defined as the nucleotide sequence of the base on DNA, which is translated into a sequence of amino acid of the protein to be synthesized. So what are the properties of the genetic code? First is triplet code. Sequence of three nucleotides codes for one amino acid in a protein, and this is called the triplet code. Second is the genetic code is non-ambiguous, which means a specific codon will only code for a particular amino acid. So it is also universal, that means so the genetic code applies to all over the species. Third is degenerate code. So every amino acid except for tryptophan and methionine is coded by various codons. So this is called a degeneracy of the codon. Non-overlapping code. The code is always read sequentially in a group of 3, and a nucleotide which becomes a part of one codon can never be part of another codon. So the codon is commaless. So that means every codon is adjacent to the previous one without any nucleotide between them. So each of them represent a codon and there cannot be any other nucleotide in between them. So we have defined start codon and defined stop codon. So ATG always is the start codon, and TAA, TAG and TGA are the stop codons. Coming to the protein expression, like in protein expression we can say like a 4-letter language, that is language of ATGC, is translated to a 20-letter language which is nothing but the amino acid during the protein synthesis. So all the genetic code is on the DNA template, and the information has to be translated into a protein. So there is an intermediate uh, molecule called mRNA. So this molecule carries all the information from the nucleus to the cytoplasm to produce the protein. So the once the mRNA is transported outside of the nucleus, so there comes the role of the ribosome, which read the nucleic acid code of the DNA and match the specific amino acid carried by the tRNA. So once the match is done, then a peptide bond is formed between the two amino acids and a polypeptide chain is formed. So once the polypeptide chain is formed, it undergoes several process of refolding and to give the functional protein. Now talking about mutation, going into the actual subject. So mutation is any alteration in the genetic material of or the genome of a cell, or it can be of a living organism. Okay, so that is more or less permanent. That means it can be a transient or it can pass to the next generation. So there are different causes of mutation. If we look into the different reason why mutation occurs, so basically three major category of mutation will find. So mutation that happens due to exposure to radiations like UV radiation or X-rays, and like if we talk about chemical mutagens like using tobacco products, benzoyl peroxidase, nitrate and nitrate preservatives, so which are found in food or some cosmetic items, so that can also cause some mutation. So if we talk about biological agent like certain viruses like HPV and some bacteria also like Helicobacter pylori can also cause certain mutations. So mutation can also occur spontaneously during like the transactions of DNA. It can happen during replication, transcription or translation. So because mutations are very random changes, they are expected to be mostly deleterious, but some may be beneficial in certain environment. So like certain bacteria get resistance just because of the mutation to certain antibiotic. So mutation is the main source of the genetic variation. So which is the raw material for evolution and for natural selection. So now let's talk about different kind of mutation. So this is called silent mutation or synonymous mutation. So for this to understand, it's very essential that you understand the codon table. So now I am taking a random DNA sequence and I'll be translating it to a protein. And suppose this is the wild type sequence, and you can see like based on the codon, so we are taking the different amino acid and just a representation of the protein here. So in case of silent mutation, so one of the codon changes like there is one point mutation and the codon changes to a different codon. But in this case, the codon when it changes, so it can still code for the same amino acid. As we know, like one amino acid can have multiple codons. So in this case, CAT, which is coding for histidine, the T muted to C. So now we have still have histidine, because CAT codes for histidine, and also the CSE also codes for histidine. So there will be no changes on the phenotypic or any there is no change in the property of this protein. So this is caused by mainly a substitution event, like the T is substituted by C. So the amino acid sequence remains the same, and the substitution can also happen on a stop codon, like suppose the TAA is a stop codon, and the A is substituted by G. So still that remains as a stop codon. So there will be no such changes in the event, like in terms of the protein expression. As you know, like the DNA is mix up of both coding and non-coding sequences, and because non-coding sequences is a huge covers a huge region in the human genome, so it is very frequently we find this substitution in the non-coding regions, and as you know, they have nothing to play with the protein expression, like they're not involved, so that will not have any kind of phenotypic effect. So now talking about the nonsense or the termination mutation. So nonsense mutation, what happens, one codon is changed in such a way that it becomes a stop codon. Like here, so in the sequence, in the wild type sequence, it was TGG, and now you can see like the G is substituted by A, and now TGG was coding for tryptophan, but TGA is a stop codon. So that means the protein expression will not proceed after once the ribosome encounters the TGA, it will not further do the protein expression part. The expression will stop here. Now this is another type of mutation called missense mutation. This is also called the substitution mutation. So in missense mutation, so in the codon, one because of one base change, like that codes for a different amino acid. For example, here the CAT is coding for histidine, but due to the substitution of one base from T to A, now CAA is coding for glutamine. So it can be a conservative substitution. That means if the after the change, the amino acid which is new, if that is similar property as the previous amino acid, then it might not have much like difference in the protein properties. But suppose if that is changed to a completely different amino acid, so definitely that is going to affect the protein property. So this the mutation where like the nature of the amino acid changes is called non-conservative missense mutation. Now we'll talk about the frameshift mutation. So in frameshift mutation, what happens? Either there is some addition of a base, or it can be two base or more than two bases. It can be three bases or more than that, and due to which there is a shift in the frame. Let us try to understand it. So this was the wild sequence, and now you see there was an addition of T here, like this was your sequence, and after 2A one there is one addition of T. Because of the addition of T, now this becomes a triplet codon, and now the A which was there, so it is now making a codon with the next C and T. So the frames getting shifted, totally shifted here. So because of this reason, you will see now the protein produced is totally different than the wild type. You see, till histidine it is fine, it is resembling the previous protein, but due to this change and the total frame of the protein got shifted. So now this person is totally a new sequence. And this is an example, like suppose in place of T, after CA there is a insertion of one amino, one codon C. So due to which you will see now, due to the shift, now this T is going to pair, making a triplet with the next two S. So because of that, that makes a stop codon. So the expression will stop here. So you can understand, due to frame shift mutation, so it can have very deleterious effect, because the protein is totally changed, or it encounters a stop codon at very early stage, so that the protein formed are very small, or these are truncated versions. So you just have to remember that even a single amino acid change can totally change the property of the protein. So now imagine, in this case, this is a totally new protein. So it may not be a stable protein, or it cannot serve the purpose of the previous protein. So this as well as this condition can bring lot of lethal effect on the cell. So these frame shift mutations are definitely one of the reason for many of the diseases. So the pre-termination of the protein synthesis, or the protein formed is a very truncated version, or a very small protein, so that is not able to give the function of the wild protein. So this is also another reason for many of the genetic diseases, the splice site mutation. Let's try to understand it. So what is you have to understand the RNA splicing. So in our genome is uh, is a combination of coding region and non-coding regions. The coding region, that means which codes for the protein, are called exons, and the non-coding region which doesn't code for the protein are called introns. During formation of the mRNA, so in the first step the pre-mRNA is formed, and this pre-mRNA will have both the exonic sequences as well as intronic sequences. So what happens in RNA splicing? In RNA splicing, there will be a role of the spliceosome machinery, so that will cut the intronic sequences so that the exon can join together to form the total coding sequence, and that will translate into the protein. But certain time it can happen, the introns the introns can be spliced out in a different fashion, producing a different protein. Now let us understand from this example. Now suppose this is exon 3, this is exon 4, and this is exon 5, and in between there are introns, and intron splicing occurs, and they have very specific sequences that can bring the splicing machinery to act, and this will have a donor site and an acceptor site. So suppose GT is the donor site here and AG is the acceptor site here. So due to certain times a mutation in the donor site, suppose this G is converted to A. So what will happen now? The spliceosome machinery cannot detect this donor site. So what will happen? So this will detect the previous donor site which is present here. That means in the splicing mechanism, so this entire portion of the this DNA will cut out. So that means, so when the splicing occurs on this sequence where there is a mutation, now the exon 3 will combine with the exon 5, uh, forming the this structure. So in this structure you can see the total portion of the exon 4 is deleted. So this is one of the reason in many of the like in many of these genetic diseases, this is one of the cause. Now we spoke about the mutation that occurs on the DNA level. Now let us just talk about the mutation that occurs on the chromosomal level. So in the chromosomal level, so there are various type of mutation, but again all of this mutation affect the total number of the chromosome, or it can affect the structure of the chromosome. So let us try to understand. So basically we will find four types of mutation in the chromosome. So one is deletion, then duplication, inversion, and translocation. So in deletion, one part of the chromosome is lost. You can see this figure, this portion is lost, and the chromosome produced will have a shorter arm. In duplication, there is some portion of the chromosome gets duplicated. Now you can see this chromosome has a longer arm here. So this is a duplication of some portion of the chromosome, like it got repeated 2 times. So in inversion, there is a like a change in the structure in the direction of the like some portion of the fragment. So it like that also changes some properties. So there is insertion. In insertion, like some person, this happens between two different chromosomes. Like some part of the chromosome can get inserted into another chromosome. So you can see it very clearly here. Now this person of chromosome 4 has inserted in chromosome 20. So translocation. In translocations, like the chromosome can extend certain portion of theirs into some other chromosome. Like here you can say this is before the translocation, and how after the translocation this chromosome has taken this portion of the chromosome 20, and the chromosome 4 has received like some portion of the chromosome 20. So these are at the chromosomal levels.
So guys, I hope you understood the topic today, and because I covered many basic concepts, and it is very much essential to understand the further session on the human genetic disease. So I hope you are going to enjoy the further sessions, and there is lot of things to learn, and also to I really, as I mentioned, I really wanted to bring a change in the society in terms of educating people on the on the genetic diseases, and how we can improve lives. Okay, so I'll be back again. See you soon. Thank you so much.